Alright, so I thought it would be a good idea to record some of these problems so that you could see how I would work them out, and it won't take up, we don't really have enough class time to go through every problem. So I'll try to solve as many as I can using this recording software. Now it's my first time through this, so a couple of these might be rough, but hopefully they'll get better over time. Even now, just trying to figure out where to put this pen is a little iffy. Alright, so I'm going to pick up a color and we'll talk about this problem. So this says give the genotypic phenotypic ratios that may appear among progeny of simple crosses, and the examples of these crosses I've shown are a monohybrid cross, a test cross involving one gene, a dihybrid cross, and a test cross of a dihybrid. So those are 4 different crosses, and those are all very specific terms. So let's start with a monohybrid cross. Monohybrid cross always means the same thing. Again, we're going to use A and A. So big A, little a, cross to big A, little a. So that's a monohybrid cross. Monohybrid cross always gives the same thing. It gives you 1/4 big A, 2/4 or 1/2 big A, little a, and 1/4 little a, little a. So that's a 4. Alright, so that's our genotypic ratio for a monohybrid cross. Now we can take these two guys. If this is typical Mendelian genetics, and it is because we're just doing the basic crosses, that means that big A is dominant to little a. So this will give you a dominant phenotype, and this will give you a dominant phenotype. So the phenotypic ratio will be 3/4 will have the dominant phenotype. So they'll have 3/4 will be that. Now this genotype can be big A, or big A, little a, so we can give it this symbol here. So this is also a genotype, a shorthand for genotype, and then 1/4 will be little a, little a. Now we're going to use this a lot. So this, if you have a monohybrid cross, you should know immediately that you have a 1 to 2 to 1 genotypic ratio that looks like this, and a 3 to 1 genotypic ratio that looks like this, where we combine these two genotypes into this form. And then this is also can be thought of as representing the phenotypic ratio. So the phenotypic ratio and we might write that just as 3 to 1. So we have a 3 to 1 phenotypic ratio. So that's a monohybrid cross. Alright, so now for the next example, it says a test cross involving one gene. Test cross is always the same thing. A test cross is always something, and this can be anything, but a test cross has to be crossed to something that's homozygous recessive for the gene in question. Now there might be 2 genes in question, 3 genes in question, 4 genes in question, but whatever, this guy has to be homozygous recessive for all the genes in question. In this case, there's only one. We call this guy the tester. If we're just dealing with one gene, we're going to get 1 AA and 1 little a, little a. So we always get a 1 to 1 ratio. Now what's cool about test crosses is that our genotypic ratio and our phenotypic ratio are going to be the same thing. So if we think about this as the genotype, and this is the phenotypic ratio, we could say the phenotype is going to be the dominant phenotype, and the genotype or the phenotype for the recessive one is the recessive phenotype. It's a 1-to-1 ratio. So both the genotype and the phenotype ratios are 1-to-1. And that's the beauty of the test cross, is that the reason that we do a test cross is so that our genotypes directly, in other words, our phenotypes actually directly reflect the genotypes. Because if you look at the outcome of the organism, you can see the phenotype, but you can't see the genotype directly. So if we do this and we use a homozygous recessive here, we know that anybody that has a dominant phenotype has to have a big A and a little a, because we're always going to get a little a from here. And if somebody has a recessive phenotype, they have to have 2 little a's. That's also a thing about recessive phenotypes in general, is that you always have to have, if you know that the phenotype is recessive, you know that the genotype is homozygous recessive. That's the definition of recessive. You have to be homozygous for this allele in order to get this phenotype. Alright, so now we're going to talk about 2 genes. Dihybrid cross. Dihybrid, that means 2 genes. Di means 2. And a dihybrid cross is very specifically something that is a hybrid for 2 genes. A hybrid means it's not homozygous. So a hybrid is another word for heterozygous. So it's heterozygous for 2 genes. So we take one of these guys and cross it to another one of these guys. So that is a dihybrid cross. It's the only thing that is a dihybrid cross. One dihybrid cross to another dihybrid. I'm not going to go through the Punnett square at this point, but if you do fill this out with a Punnett square, and maybe we'll do that in a second, you always end up with the same ratio. But right now I just basically want you to memorize this ratio, and you've all seen this ratio. 9331, the infamous 9331 ratio. But we're going to draw out the genotypes like this. So 9 of these guys will have the dominant A phenotype and the dominant B phenotype. But of these, all the ones that show these dominant A and dominant B phenotypes, so we'll put the phenotypes over here, dominant A phenotype, dominant B phenotype. The A can be big A, or big A, little a. So we give it this little symbol, and B can be big B, or big B, little b. So we give it this symbol. Now 3 of these guys are going to have the dominant A phenotype, but the recessive B phenotype. There's only one way to get the recessive B phenotype, and that's if you're homozygous for little b. And so this would be the dominant A phenotype and the recessive B phenotype. So this is just a shorthand for the phenotypes that we're talking about. Now the thing with genetics is we use a lot of these A's and B's, and sometimes they mean different things. So you have to get used to sort of A's and B's and different letters being abstract symbols to represent what's going on at the genotypic level. Now these 3 we're going to have be recessive for the A phenotype, dominant for B, and we'll have that kind of phenotype. And one of these will be recessive for both. So that's the infamous 9331 ratio of a dihybrid cross. We could go through this on a Punnett square, and maybe we'll do that in a minute. But just remember this ratio. Now another thing to think about these is to think about them in terms of fractions. So this is 9/16, 3/16, 3/16, and 1/16. And so that becomes useful when we start doing probabilities. When you want to calculate probabilities, you need to use fractions to represent a probability. So each of these things represents the probability of getting this type of offspring from this kind of cross. So if you're given a dihybrid cross, the probability of getting something with both dominant phenotypes is 9/16. So we'll come back to that later. Alright, so we can get rid of this little layer and do another one. So now the next one is a test cross of a dihybrid. So that's very specific terminology, a test cross of a dihybrid. We know a dihybrid is this, and we know a test cross is always homozygous recessive for all the genes in question. So now that's a test cross, but it's a test cross involving two genes. And so this will give you this ratio every single time. So phenotypically, what you get is a big A and a big B, big A and a little b, little a and a big B, little a and a little b. And we're going to get this in a 1 to 1 to 1 to 1 ratio. Or 1/4, 1/4, 1/4, 1/4. Sorry, I'm still getting used to this pen. Alright, so this is the phenotypic ratio, where these are just the abstract representation of what the phenotypes are. Dominant for the two big, or the two, showing the two dominant phenotypes, dominant A phenotype, recessive B, etc. What does that look like in terms of genotype? Well, as we can see, we can get this allele from here, and we can get this allele from here. And if we do that, the only thing we can get from here is little a or little b. And so we can always only get little a or little b from here. But from here we can get, if we get a big A, we can get either a big B or a little b. So we can get a big A and we can get a little b. As we'll see later, that's what we mean by independent assortment. Big A can go with B, or big A can go with little b. But from the tester, we can only get little a, little b. So this guy is always going to give us the same thing. Now from this parent, we can get a little a, and we can get this big B. But we always have to get little a, little b from the other guy. So that's these guys. And we can get little a, little b from this parent, and always get little a, little b from this parent. So as we'll talk about as we go through meiosis and everything, these types of things that we get from this parent are always going to be equally likely. So we can get big A and big B 1/4 of the time, big A and little b 1/4 of the time, little a and big B 1/4 of the time, little a and little b 1/4 of the time. And because this is a test cross, from here we can only get little a and little b every time. And so that means little a and little b, they don't contribute to the phenotype because they're recessive. So whatever phenotype we get, whatever genotype we get here, so if we get a big A to big B from this parent, that means we'll get the big A and big B phenotype. So there's a direct correlation between the genotype and the phenotype. As you saw with the dihybrid cross, if we had this being big A, big B, little b, sometimes we'd get a big A from here, and sometimes we'd get a big A from here. And if we had a dominant A phenotype, we wouldn't know if we got two big A's or a big A and a little a. So that's pretty much this problem, but let's summarize it. Oh, I think I put that on the background layer. Alright, so I want to get rid of all this stuff. Let's see if that's possible. Rid of it, but we also got rid of the black stuff. So let's put the black stuff back in there. Alright, well, it's not quite black, but it's close enough. Alright, so now we can summarize this stuff. So we get a monohybrid cross, get a 1 to 2 to 1 ratio or a 3 to 1 ratio. Test cross with one gene, 1 to 1. Dihybrid, 9 to 3 to 3 to 1. Test cross, 1 to 1 to 1 to 1. so these guys are important. So whenever you see these ratios, you know if you see 1 to 2 to 1 or 3 to 1, you know you're dealing with a monohybrid cross. What does that mean? That means we're dealing with one gene. And it's this kind of a cross. 3 to 1, it's a phenotypic ratio from this kind of cross. 1 to 1, that means we've got big A, little a cross to little a, little a. So if you see that, that's what we've got going. Now of course it's not always going to be the case, but that's where we start, is that we're going to look at a cross, and our first question is going to be, does it fit these typical Mendelian ratios? Dihybrid cross, 9, 3, 3, 1. Test cross, 1, 1, 1, 1, 1. 4 of those. Now don't assume it's 9 to 3 to 3 to 1. The only time we have 9 to 3 to 3 to 1 is if it's a dihybrid cross. So those are the kinds of